So how do bacteria grow? Well, like most things, you need food, okay? So food for bacteria are nutrients of various kinds, but sugars, amino acids, vitamins, nitrogen, phosphate, all the stuff that everyone needs. This food, these nutrients, are converted into macromolecules. Macromolecules include RNA, DNA, and proteins. Macromolecules then are produced to make up a new cell. So you need all these macromolecules to make a copy of the cell. If we look in more detail, as shown here, here we have a cell. That's the DNA there. That's a plasmid. What happens is the DNA is going to copy itself. So it completely copies the DNA into a new circle of DNA. This is happening while the cell is getting longer and longer. So the cell is getting longer and longer and longer. The DNA then is pulled to either side of the cell, and a division septa is put in the middle here. That septum then forms and creates new cell wall and new membrane between the two new cells, and then the cells divide. So it's essentially they're making clones of themselves. Every new cell is identical. This shows a picture, a real picture of bacteria that are dividing. This is E. coli. You can see here, this cell is about to divide. You can actually see the septa in the middle. It's about to cut in half the cell. An even closer look is here. Here's the E. coli cell that is about to divide. You can see the septa here. These white blobby things are the DNA, and all the little dots are ribosomes. Now, exponential growth is a concept also called log growth. If you think about it, one bacteria becomes 2, 2 becomes 4, 4 becomes 8, etc., etc., etc. That's exponential growth. And to give you a sense of how quickly this can happen, I might be able to show you a video, or not. So this shows bacteria growing on a slide under the microscope. And it is sped up, of course, but it's still incredibly fast within a few hours. Specifically, a bacteria like E. coli can grow every 20 minutes. That's why I always mention E. coli, because it grows so fast and so easily that we have a non-dangerous form of it. We can use it in a lot of experiments. So if you think about that, in less than 7 hours, one E. coli will produce 1 million daughter cells, because 1 becomes 2, becomes 4, etc. Other bacteria grow much more slowly. For example, Mycobacterium tuberculosis only divides every 24 hours. So how do we grow bacteria in the lab? There are different ways, but one of the ways we work with them is by streaking them on plates. I'm going to show you this. So this here and this here is an agar plate. The agar plate is like a gel, a semi-firm gel with food in it, basically, for the bacteria. When we want to work with bacteria, we often want to purify them. Let's say I take a sample from Martin here. I might get 100 different kinds of bacteria. So usually what I want to study are individual species of bacteria. And to do that, I streak them on the plate using a loop. Streak them here, and then sterilize the loop, or take a new one, and streak through the initial one, take a new one, or a sterilized one, streak again, and streak again. What happens then is I'm diluting out the bacteria as we go along. So I start out with a lot of bacteria, and here very few. Each of those little colonies, they're called, started with one bacteria. So in a typical experiment with E. coli, this I would leave overnight, and I would see the next day very visible colonies containing one bacteria that grew up into about 1 million or 10 million. So that's one way we work with bacteria in the lab. The other thing to keep in mind about bacteria is that bacteria divide asexually. Eukaryotes, like people, require a sexual phase. So you have male and female. Your offspring is a mixture of the genes from both parents. Bacteria don't have this. Bacteria basically are clones. They divide in half. Every clone is identical. So when I streak them on this agar plate, I end up with a lot of clones, all identical. In addition, bacteria are haploid. What that means is haploid is only one copy of the chromosome, and therefore one copy of every gene is found in a bacteria. On the other hand, a diploid like you have 2 copies of the chromosome, 2 copies of every gene. You get one from your mother, one from your father. Bacteria don't have that. They have 1 copy. Okay. So before I take questions, if you wanted to kill a bacteria, what do you think you could do? What could you target or inhibit? If you think about the cell's growth and structure. And in the next section, I'll tell you how some of the antibiotics work. So discuss for a minute.